everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this forensic science lecture series, today we will be talking about collection, packaging and forwarding of physical evidence. In the last series lectures, we have learnt about various physical evidences which can be found at the crime scene. Now, whenever there is some crime reported, there is a mobile forensic science lab which goes to each and every crime scene. This mobile lab has various investigators' kit, various chemicals which can be used for the analysis of evidences that can deteriorate very easily due to the environmental conditions. This mobile forensic unit is available at each and every forensic science lab and CFSLs, and these mobile forensic science lab goes to each and every crime scene containing all the important material for the collection, handling, packaging and transferring of the evidences to the various forensic science lab meticulously. So if we see the investigator's kit consists of the mask, the gloves, it consists of UV light, magnifying glass, scaling, tape, various cutter, evidence sheet, evidence number and various types of investigating kit which helps in the collection or the handling of evidences. Before collection of evidence, we should take in mind various precautions. Always use gloves. Each investigating officer never touch any physical evidence using bare hands. He or she should always wear glove. Any object which might contain any latent evidence, for example, if we consider any case of murder, there could be latent fingerprints which are present on the edge of the weapon. For example, if the weapon is a knife or if the weapon is some sort of firearm, there could be latent fingerprints which are present on that firearm or that knife. So the investigating officer or the officer who is collecting the evidence should hold the object or hold the evidence in such a manner that the fingerprints which are present on that evidence should not get damaged or should not get wiped off. The most important point to keep in mind is not touch anything with the bare hands, and if the investigator is taking round of the whole crime scene, his shoes should be covered, his body should be covered and his gloves should be covered, because he should not leave any extra evidence on the crime scene of his own. If he does not cover his foot or his shoes, then he might leave his fingerprints on the crime scene or footprints on the crime scene, or if he is not wearing gloves, his or her own fingerprint might get transferred to any surface, and that might create a confusion in collecting the evidence or at the crime scene. So, before touching any object, the IO should wear gloves. On all individual packets and as well as in the collective packet should contain the signatures of the witness and the signature of the IO. Each and every evidence which is collected should contain the signature of the witnesses who are present at the crime scene and also the signature of the investigating officer. The investigating officer should decide to pack one exhibit at a time. For example, if we consider a crime scene and there are certain evidences, the investigating officer should pack such evidences before which can destroy or deteriorate with environmental factors. For example, or any other evidence through which the loss of evidence is possible. For example, if we consider a case of a murder and there is a knife where latent fingerprints are present on the knife, the investigating officer should pick up the knife first and should pack the knife in such a way that the fingerprints which are present on the knife should not get destroyed or the evidence should be kept intact. Each and every detailed description of individual physical evidences such as the type of object, make, color, number, dimension, location, etc. Whichever is applicable, the detailed description should be given on that evidence collection sheet. For example, if a fingerprint is being collected, the investigating officer should tell that this particular fingerprint is collected from this particular table, and the multiple locations, if the multiple locations are there, he should mention each location number wise, that this is the fingerprint from the location 1, this is the fingerprint from the location 2. Similarly, if there are multiple blood spots at the crime scene, the investigating officer should never collect all the blood spots together, because they might belong to different persons. So he should look carefully and see that if the blood spots are at various locations, he should name each and location. This is location 1, this is location 2, this is location 3, and from each locations the blood should be collected in different paper bags or whatever the appropriate material is. He should always consider any blood-stained object such as dagger, lathi or bullet should be dried first and then packed. Whenever we have any wet blood, it should always be air dried, not in sun, not in heat, because that will destroy the cells, and that will we won't be able to extract DNA out of that evidence. If there are certain trace evidences which are being stuck to the weapon or to the murder weapon, then they should not be removed. For example, if there is a murder weapon and there are certain hairs and fibers which are stick to it, the investigating officer should not remove those hairs and those fibers. Instead, those evidences shall be packed with such trace evidences properly in the paper bag after air drying it. All the biological stains should be thoroughly dried under shade. They should never be dried under sun or intense light, reason being that the rays of sun will destroy the biological evidences and then it can deteriorate the DNA. It would be difficult to extract DNA out of those samples. So, the wet samples should be air dried either under the normal temperature in the fan, but never be dried in the sun or intense light. Under no circumstances, biological stains should be packed in airtight packaging materials, for example cellophane, polythene or glass vials, because the biological stains might give rise to certain microorganisms, and these microorganisms may result in the deterioration of the evidences. So, the biological stains should be packed always in non-airtight packaging materials. Physical evidence should be packed separately without mixing or contamination. Each and every physical evidence should be packed separately. In fact, if the physical evidence, for example, if we have talked about the blood, the same physical evidence which is found at the multiple locations should be packed separately. If you have found certain paint chips at different locations, you should pack those paint chips from different locations in different packagings, because they might belong to different surfaces. So, the forensic lab scientist then further can examine whether they belong to the same location, or the evidences which are being collected from different locations, they have the same source or not. But the investigating officer who is present at the crime scene should collect all the physical evidences separately from multiple locations. Every item should be protected adequately with suitable packaging material against external shocks so that they do not break during the transit. Let's consider a case of a firearm. If you have a firearm, the firearm should be packed, or any evidence, for example the bullets are there or the cartridges are there, they should be packed properly in a box, and along in that box you should add some pieces of cotton or some thermocol material so that the evidence packing in that box is tight. It should not move here and there, because the transit may give certain marks to that bullets or the cartridge cases or the firearms, which may hinder while you are evaluating the evidence, because the lines which are present on the bullets and the cartridge cases, they are the mark through which you identify whether they belong to the same weapon or not. So, any evidence should not get any extra lines or any extra shocks. For that, to prevent that, it should be packaged very carefully with cotton or with thermocol so that it should not move during the transportation. Also, to protect the latent, visible or plastic fingerprints on the object, they should be packed in such a manner that the packing materials do not encounter the object. They should be covered properly so that if they are packed in a paper bag, it should not touch the paper bag. Finally, all the individual packets of physical evidences should be packed together in a box, wrapped properly using intact piece of paper or cloth, tied and sealed. All the evidence from a single crime scene should be packed separately in different packages, and then together they can be packed in a big paper bag. Now, when you are transferring those evidences, there should be a forwarding note that will help the person who will receive the evidences at the other end. What is the other end? That is the forensic science lab. We usually make 3 copies of the forwarding note. The first copy should be placed in sealed envelope inside the box. Second copy should be handed over to the messenger in a sealed envelope along with the box containing physical evidence, and the third copy should be retained in the case file of the police station. So the 3 copies which are being made, one is inside the packaging material, second is sealed in the envelope and given directly to the person who is receiving the evidence, and the third copy which is retained at the police station. As you can see in this picture, the seal, the red color seal which is present there, it should be marked. The seal which is used by the investigating officer, the sample of the seal should also be present in the box or in the paper bag which is being given to the forensic science lab authority, so that if during transit someone tried to hamper or tamper the evidence and they might have changed the seal, the person who is receiving, he should know that this is the way the seal looked like. So they should check from the sample, yes, our seal is intact. So they will match the seal with the which is inside the package with the seal of the envelope they have received. This is very essential because we have to see the proper chain of custody is being maintained. Chain of custody, as we talk about, we say the evidences are being collected by the investigating officer, then they are transferred and being sent and received by the FSL member. So in the whole process in between, if someone tried to hamper the evidences, the person will get to know that at what point the seal was broken, and then he can be accounted for the same. All the individual packets of the exhibits, control samples and copy of the forwarding note should be placed in a suitable box, wrapped with intact piece of paper and sealed. Also, we should mention the name of the addressee and the address clearly along with the FIR and the crime number. Whichever box in which we are sending all of our samples, it should be properly mentioned that this package is going to particular forensic science lab and it is being sent by this particular police station or this particular police officer along with the FIR number, the case number, the proper FIR number, case number and the investigating officer signature and the forwarding slips. Everything should be mentioned there along with the package. All the individual packets of exhibits, control sample and the copy of the forwarding note should be placed in a paper bag or a suitable container, properly closed, tied with cloth tape or twine, and the seal, they should apply seal wherever required to ensure that the exhibits are not tampered. As you can see, there are multiple packaged evidences which are being packed in a big box, and on that box they have pasted one forwarding note as well as the sample of the seal. As I have explained that whenever required, the exhibits are not tampered, the intact of the seal should always be maintained. This picture is showing the final packet of the exhibits and the control samples handed over to the messenger who will deliver them to the forensic science lab. So it contains the proper name of the addressee as well as the addressed and the seal, and the same seal sample is also present inside the box, so that the person who has received should match that the samples are intact and the seal is also intact. Messenger will personally take the parcel and hand it over to the forensic science laboratory for examination. The person who is sitting at the end, who will receive the sample from the packet from the messenger, he will check all the necessary information, the forwarding note, the forwarding seal, the signature of the IO, and he should also check that the sealing should be done properly and should remain intact till the packet is handed over to the forensic science lab or the central forensic science lab. The messenger hands over the exhibit and the forwarding note to the sample reception unit of the FSL, and the person at the reception examines the seal whether they tally and intact. They look for the signatures of the IO, each and everything, they matches the signature, and then when they receive the packet, they give a receiving receipt to the messenger that we have received this packet, and he will take that receipt to the police station who has sent those packet or the exhibit to the forensic science lab with the signature that we have received the samples with the seal intact. That completes the chain of custody. He then opens the forwarding note, reads the contents, opens the exhibit bag, verify the article mentioned in the forwarding note. The forwarding note should contain each and every article which is being packed. Like the first packet contains some murder weapon, second contains the fingerprints which were lifted, third contains some blood samples, fourth contains some physical evidences of physics department. So he will tally each and every one, and he tallies with the second copy of the forwarding note which is signed and returned to the messenger as a receipt. The exhibit reception unit of the forensic science lab will acknowledge the receipt of exhibits and the copy of the forwarding note, and they hand over to the messenger. The receipt of the exhibit is placed in the case file of the police station, where they will mention that we have sent these particular exhibits and samples to the forensic science lab. Most importantly, chain of custody of the exhibits packet should be maintained throughout. That is, from the collection to handing over to the FSL until the trial is completed. Now we move towards how the various physical evidences which are present at the crime scene are being collected. There could be many evidences which are found at the crime scene. It could be, most importantly, the first biological evidence which is being encountered and which is a major evidence which can help in the identification of the victim as well as in the identification of the suspect is blood, because through biological evidences we can extract DNA. Other evidences which can be found are murder weapon, and if the person who has committed the crime using a firearm, we can find the GSR. In the cases of any fire explosive or arson cases, we can find various any bomb explosive devices or any materials which help in the result of fire, or we can have some files or some documents in the case of question documents cases. Mostly we can find the fingerprints, footprints, etc. at the crime scene. So now we will discuss each and every evidence, how they are collected at the crime scene meticulously. The most important evidence, as I say, are the blood sample or the body fluids. So the blood samples can be found in various forms at the crime scene. It could be in the form of liquid, and it could be a pool of blood. It could be a dried sample. The liquid sample is when the crime scene is fresh, and the dried blood samples are there when the crime scene has it's been 2 hours, 3 hours, or a few days when the crime is being happened. So the collection of the evidence depends on what type, in what form you have found that particular evidence at the crime scene. So if you have found the blood in the liquid form, it can be collected on a sterile cotton swab, and the cotton swab should be air dried before packaging. If there is a pool of blood or if there is any suspect, then their blood sample could be collected in the EDTA vial directly. For the dried blood stains, as we spoke previously also, that all the biological materials which are in wet condition, they should be air dried first, and the dried blood, it should be photographed first, and the investigating officer should also document the pattern with the scale prior to collection, because the blood stain pattern helps us in the reconstruction of the crime scene. The preferred method is to send the entire article containing the stain to the laboratory. Supposedly, if your crime scene contains dried blood stains on clothes or any other thing which can be directly packaged, then you can send the whole article to the crime scene. But if it is on the wall or if it is on any big surface like any furniture, in such cases you should scrape the stain and you should pack it in a druggist fold or use a sterile cotton swab moistened with sterile distilled water and then air dry it and then pack it. Now, what is a druggist fold? Basically, it is if we take a A4 size sheet and then we make it a 3-fold. After making it a 3-fold, in the middle of the fold, you keep the dried samples. Basically, all the dried blood stains, all the trace evidences, paint samples, glass sample, etc. Druggist fold is being used for the collection. After making 3 folds, we cover from the left hand side and the right hand side, and we put the right hand side in the left hand side and make the druggist fold intact. Then this druggist fold can be tied with a twin, tied with a thread, and then a seal can be made on the top of the druggist fold. So the dried blood stain, if as we discussed, the material is quite bigger, then you scrape the stain and then the scraped part can be put inside the druggist fold and can be properly threaded and it can be properly sealed. If there are any blood-stained weapons or clothes, hold the object carefully so that fingerprints are not wiped off. This is a very important point which is to be taken care of, because mostly murder weapons can have the fingerprints of the suspect or the person who has committed the crime. So, the murder weapon or any blood-stained weapon or clothes should be held very carefully. Do not remove any trace evidences from the blade of the knife, because the whole evidence should be packed as such. It is the job of the FSL scientist to take care of those evidences together, because in the process of removing the trace evidence, you might lose any specific or any hair root bulb which can result in the extraction of DNA. Any wet blood should allow to be dry completely in the air only, and then we can wrap the blade or the knife with a clean paper. As you can see in this image, that a knife the blood-stained knife is being packed in a paper. It should be completely packed and the person is holding the back of the knife with not with the bare hands, with the gloves, so that the fingerprints are not wiped off. And we should place the thermocol on both the ends of the exhibit and place it in a cardboard box in such a manner that the exhibit does not come in contact with the packaging material. We should close the box and ensure that no damage is caused to the exhibit or contaminated during the transportation, as we have discussed earlier about this. It should be wrapped properly with intact piece of paper, preferably brown paper, so that it can be tied and the seal is clearly visible on this paper. Seal the ends with the adhesive tape, twine as shown in the picture. After mentioning all the details, the FIR number, type of exhibit, etc., on the top of the brown envelope, you should also mention the forwarding note along with the seal. This will directly go to the forensic science lab and they can see which all evidences are being packed and how the different dried blood stains are being packed in the proper packaging material. So, in the next series, we will discuss the collection of more evidences and how those further evidences can be transferred to the forensic science lab, maintaining proper chain of custody. Thank you.